479, 279. So these questions here. So these 9 questions. So let's take them up. I don't know if you've done them or not, but regardless, we'll take them up. Okay, so question number 1 asks, what would Hershey and Chase have concluded if they had found radioactive sulfur instead of radioactive phosphorus inside the infected bacteria cells? Yeah, Du was right. The answer that she has in the chat is basically the correct answer. It would have indicated that the protein was a genetic material, not the uh, DNA. Question number 2. Nitrogenous base content of a sample was found to be 32% adenine. Determine the amounts of the other 3 bases in the sample. So if adenine is 32, then thymine has to be 32 because A has to equal T. And then if you do 50 minus 32, that gives you 18% for guanine and cytosine. Okay. 3, describe and sketch. Well, you can't really sketch unless you're going to show me a picture, but describe the structure of DNA. So, to be honest, this is probably the best example. If you look at figure 7 on page 4278, there's a good picture of DNA. So that's basically question 3. Question 4. Write the complementary sequence for the following sequence. So G goes to C, T goes to A, G goes to C, A goes to T, G, A, T, T, G, T, C, A, C, C, G, G, T, A. If you need me to repeat that, let me know and go through it a little more slowly. Yeah, that looks right. In point form or notes or a timeline, summarize the findings of. Okay, that's basically, in fact, if you look at my last slide for today, from yesterday rather, once it loads, that's it. That's the timeline. And that should be. Okay. So that's basically the timeline in terms of the significance of their findings. Okay, question 6. Distinguish between. Okay, the nitrogen base is just the single or double rings. The nucleotide includes the sugar and the phosphate. Okay. Purines are adenine and guanine. Okay. Pyrimidines are thymine, cytosine, and uracil. And Let's see. Yeah, A and G are the double rings, and T and C are the single rings. Okay. Number 7. How did the reproductive behavior? Oh, bacteria and bacteriophage. Sorry, bacteria is a prokaryote. Bacteriophages are viruses. Number 7. How did the reproductive behavior of bacteriophages allow Hershey and Chase to conduct their experiment? Because bacteriophages are viruses, so they inject their DNA into cells. So it tells you whatever gets injected into the cell has to be the genetic material. Why were Avery, McCullough, and McCarty hesitant to report their findings? Um, because it was possible that if they didn't destroy all of the protein, some of the protein could have contaminated their results and ended up giving the results as they saw. Because most people thought that proteins were the biological genetic material. Why is it important for science? Because scientists basically build on both their findings and on their errors. Watson and Crick did not actually conduct any experiments. Do you think they can be considered scientists? Explain. Okay, that's an opinion piece. Question 9. So long as you can support your answer logically, I would accept it. There is no right or wrong answer for question number 9. Okay. How many of you think that they are? What's the question? Are they scientists? Was that the question? Yeah. Okay. How many of you think they're scientists? Hands up. Okay. How many think they are not? Hands down. How many don't know? Yeah. It's an interesting question. Is a theorist a scientist? That's what they're basically saying. And if that's true, well, let me ask you this. Is Einstein a scientist? Yes or no? Hands up if yes, hands down if no. Okay, because Einstein was a theorist. He never conducted one physics experiment. He couldn't. He had mind experiments. Yeah, he was essentially a mathematician. But people still call him a scientist. Is he a scientist? It's more of a philosophy question than anything else. There is no right or wrong. So as I said, as long as you can support your answer, I will give you credit for that question. It's one part of it. It's not all of it. He had an element named after him, sure, Einsteinium. That's true too. I mean, if you compare Einstein to someone like Rutherford, right? Rutherford actually conducted experiments. No, Abraham, he didn't discover Einsteinium. I think there's a general rule in chemistry that you cannot name an element after yourself. So Seaborgium, Californium, um, it's a UPAC, I believe, that actually names the elements, which is why the last 4 elements were named about 3 years ago. Yes, that's why that's why Lawrencium is um, one of the is named his name was given to one of them. But yeah, you're right. Berkeley did a bunch as well. right? Okay, so let's continue on. Finish. So we got to the point where we talk about DNA. What I want to do now is we were here, where we were looking at how DNA replicates itself. So how is DNA copied, right? I think I showed you this slide. No, that's the last slide I showed you. Okay, so moving on from there, the question is how is DNA copied? Okay, so the replication of DNA, okay, base pairing, right? A pairs with T and G pairs with C, says that each side can act as a template. So each can guide the new side that's being made. Okay. So if you look here, the blue strand is the old strand, and it dictates what's going to go in as the new strand, which are the gold ones. Okay. So as Watson and Crick even noted, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. Okay, so we know because of base pairing, as long as you have one side, you can make the other. The question is, how is that modeling done? There are basically three ideas. Okay, the idea one is called conservative replication. Okay, and in conservative replication, when you make the new strands are made, and then the new strands pair up, and the old strands pair up. Okay, and then that happens again. So the new strands are 100% both new strands, and the original strand stays together. Okay, that's what we call the conservative model of replication. The second model is called semi-conservative. You start with the original strand, they split up into two, and each original strand builds a new strand. So they're 50% original and 50% new. And this keeps going. Okay. The last one is what's called dispersive. So you get a mixture in each strand of both old and new. Still ends up being about 50-50 in each strand, but each strand consists of both old and new DNA. Okay. So can you design an experiment to verify? Okay. So these are experimental predictions, which means these are what? What's another term for an experimental prediction? Starts with H. Yeah. So we have three different possible hypotheses for how DNA can replicate. Okay. Uh, the experiment gets fairly detailed. So let's say we take a 10-minute break right now, and we'll start again at 10 o'clock. Okay. See you then. Okay, so let's look at the experiment. So the first line obviously is labeled P. This is the parental generation. 1 is the first generation born after the parents, and 2 is the second generation born after the parents. Okay, so how do we actually do this experiment? Okay, kind of giving you the answer here. This experiment is the semi-conservative replication experiment done in 1958 by Messelson and Stahl. Now, this time, again, they're using a element um, that uh, gives off a signal, okay? And the element they're using this time is nitrogen. Now, in the parent generation, what you have to do is you have to have every single nitrogen in the DNA be replaced with this new nitrogen. So this is nitrogen 15. Okay. Normal nitrogen is nitrogen 14. So what's the difference between nitrogen 15 and nitrogen 14? Okay, so nitrogen 15 has one more neutron. So its atomic mass is one atomic mass unit heavier, which is why it's nitrogen 15 instead of nitrogen 14. So you're going to incubate the E. coli bacteria with in a solution containing nitrogen 15 so that all of their DNA incorporates the heavy nitrogen. Okay? And you're going to do that, let that sit maybe 24 hours so that the entire population has heavy nitrogen in their DNA. There is no other nitrogen available to them. So all the regular DNA is now gone. It's only heavy nitrogen. What you then do is you transfer the bacteria into a new solution containing light nitrogen, nitrogen 14. And you put them into that new solution for 20 minutes. Okay. After 20 minutes, you're going to take a sample out of the bacteria. You're going to break those bacteria open and you're going to centrifuge that solution after 20. You're going to centrifuge that sample. The DNA is going to end up in a pellet at the bottom. Okay. You're going to let this solution continue replicating and continue running for another 20 minutes. So after 40 minutes, you're going to take another sample, break it open, and centrifuge it and collect its DNA. Okay, why 20 and 40 minutes? Yeah, bacteria replicate on average at about every 20 minutes. So if you do that, you know that there's only been one replication here, whereas in this sample, there will be two replications or two generations of replications done here. Okay, so we're now going to make predictions. Okay, in red will be the N15 parent strands. Okay, and since all the nitrogens are N15, the DNA should settle out at one location. Okay, so this is where the N15 lines are going to be. Now, oops. Okay, in conservative replication, okay, So in other words, if old strand stays with old strand and new strand stays with new strand, right? When you look, what you should get is one line at N15 and one line at N14. Why? Because okay, if we go back to slide 22, right? There's a dark blue, which are N15. You should get one strand that's completely N15 and one strand that's completely N14. So in round one, you should see two lines. What should you see in the first round of replication under semi-conservative? One or two lines, first off. Okay, so if you go back, one strand, one half of one strand will be old, one half of one strand will be new. So that's a combined in 1415. This will also be a combined in 1415. So what you should actually see will be one line and 1415. What about the last one? When it's dispersive, what should you see? Yeah, because they're going to be combined, should be one line. Okay. All right. Under conservative replication, what should you see in your after 40 minutes? Okay. Same as before. So you should see that. What should you see under semi-conservative? Definitely you're going to get 11514, but let's take a look. Here's semi-conservative. So there's your 1415 and there's your 1415, but what are these two going to be? These two should be pure 14s, so there should be two sets of lines. Right? N14, 14, and then 15 and 14. Then the last one should be like the first one, 15, 14 combined. Okay? If you do the experiment, okay, there's the parent, a really strong N15 line. There's the first generation, a really strong N14-15 line. So that means the conservative is out. Round two, we'll get an N14-15 line, an N14-14 line, which means that one's out and that one's out. That's the only one that's available. So this is considered to be one of the most elegant experiments done in biology. Okay, so yes, Grant, it was semi-conservative. Oh, this is Messelson and Stahl. This was them when they were at Harvard at a Halloween party. Messelson and Stahl are those two there. Typical grad students. You're drinking lots of beer. So this basically summarizes the timeline for understanding what the DNA is a genetic material. Okay. So Kossel said that the 5 nucleic bases are A, T, G, C, and U. Morgan said genes are on chromosomes. Levine says that bases are sugar, phosphate, and nitrogenous bases. Griffiths says that some transforming factor can change phenotype. Avery, McCarty, and McLeod said transforming factor is DNA. Jargaff came up with his pairing rules. Henry and Chase confirmed DNA is a genetic material. Watson and Crick figured out the structure of DNA, and Messelson and Stahl figured out how replication itself worked. Okay. One concept I want you to understand, because we're going to be talking about this quite a bit throughout the rest of this unit, is something called the central dogma. Okay. It's how genetic information moves inside a cell. Okay. DNA can either copy itself, which is replication, okay, or it can be used to make proteins. And it does so by copying itself over to RNA, and that's called transcription. And then the RNA is turned into protein, and that's called translation. This route, DNA, RNA, protein, is the central dogma of genetics. What's a dogma? Yeah, it is the central belief of genetics. Okay. The problem with dogmas is oftentimes they're shown to be not always 100% true. Okay. Turns out there are cases where you can have genetic information coded as RNA, and then in some viruses, and then it'll go back to DNA once it infects a host, and then go through the central dogma. Okay. Any questions on this? Okay. I'm going to give you an assignment. It won't be due for at least a week. But the basic assignment is this. I want you to go to the following website, which is in Google, search for DNAi, which is the website DNA Interactive. Okay. And I want you to go to timeline. And what you will see here is a timeline from the pre-1920s to the year 2000 on the discovery of DNA. So a variety of people involved, right? There's Gregor, Meichner, Cornus, De Vries, von Seineg, Thomas Hunt Morgan. 1920s, we've got Mueller, Barbara McClintock, George Beadle, Edward Tatum, Letterberg, Avery, Wiltkin, Chargaff, Franklin, Chase, Hershey, Pauling, Crick and Watson, Bosner, okay, Crick again, Zarmanek, Hoagland, Messelson, Stahl, who we just talked about, Kronberg, Brenner, Nuremberg. Okay, the reason I'm showing you this, we've taken genetics to about the late 1950s, right? These are the people we've basically discussed. What we haven't done is looked at the 1960s, the 70s, the 80s, the 90s, and 2000s. Okay. What I want you guys to do, pick someone from 1960 through to 2000. I want you to give me, pick a scientist, pick one of these researchers. I want you to give me their biographical background and what their contribution was to the field of genetic studies. What did they discover? That's the assignment. Okay, so let me put that up on classwork. Genetics biography / contribution. Oops. We'll talk about it in a minute. Let me set up the assignment. Researcher from 1960s through 2000. And yeah, present. By present, I mean write a paper. Um, okay. Um, include a bibliography. Yay, Chicago, MLA. You don't necessarily need in-text citations, but I do want a bibliography. What else? How long should it be? Minimum 1 page. Spaced. Maximum 3 pages. I don't want to read really long things, 7 pages long, so no more than 3. I'm going to stop reading at 3 pages. So if you have a lot of information, figure out what's really important and prioritize your information. Minimum smallest font size 10 point. No unreadable font. I did have someone submit a paper in Wingdings once. Just a doc. I mean, if you want to do a slide, you give me a slide presentation, sure, but then no more than say 5 slides, 5, 6 slides. I think a paper is easier. If you want to do something other than a doc, talk to me about it. Okay. Topic 25th to 28. Points, it'll probably be out of 10 total. Due date. Okay, today's the 27th. You guys have Relay for Life tomorrow. So I'm thinking, how about a week tomorrow? Is that enough time if I make it due the 4th? Does that sound reasonable? Sure. Okay, so call it the 4th. If you want to shift that later, like next week after thinking about it, or later in the week, let me know. We can always change it. I think that's it. Assign. Okay, so that's there. The other thing I'm supposed to do, I just got the email. Where is it? Oh, that's why. Okay, this is the last promotion for Relay. So I'm supposed to show this to you.
OCDSB Relay for Life is this Friday at 2 p.m. Get excited for a jam-packed event full of exciting speakers, prizes, games, and performances.
We even have hundreds of dollars worth of prizes available for people in attendance. And don't forget that just by signing up and attending the event, you are eligible for 2 volunteer hours.
If you haven't signed up for the event yet, no worries, it is not too late. You can still sign up at relayforlife.ca/OCDSB, and it'll only take 2 minutes of your day, so go do it now.
You don't want to miss out on this awesome opportunity to help make a difference in the lives of Canadians facing cancer, as well as earn volunteer hours, win prizes, and have tons of fun. Speaking of prizes, another great chance to win some will be this Wednesday after school during the Relay for Life scavenger hunt. Check out the Student Center this Wednesday at 3 p.m. for your first clue.
And just a reminder that for fundraising, you can gain an additional 3 volunteer hours, totaling at 5 hours if you also come to the event. And if you haven't had the chance to fundraise yet, I challenge you to take a few minutes out of your day to help us reach our goal of raising $25,000 for the Canadian Cancer Society.
The top 3 fundraisers will each win $50 gift cards, and anyone who raises $50 or more will be put into a draw to win a $25 gift card. So get excited, OCDSB. Sign up today and fundraise at relayforlife.ca/OCDSB.
Okay, so that's that. Okay, so the next bit that we're going to be doing classwork, is DNA replication. So the slideshow you want is this one here, 03. Hopefully this loads soon. Why is that last picture not showing up? Oh well. I assume you the presentation's on your screens now. Okay, we'll go for 20 minutes. Okay, so as we've said, Watson and Crick figured out the structure of DNA with the double helical structure based on the work of other scientists. Okay, so as they said, right, this base pairing suggests a possible copying mechanism for genetic material, which Messelson and Stahl basically managed to figure out. Okay. Now, question then becomes, how does DNA actually replicate itself? Okay. Well, a couple of things we have to talk about first before we talk about DNA replication is the fact that DNA has directionality. Okay. Which means, again, like in sugars, we have to number the carbons. Now, when we're numbering the carbons in the ribose sugar, okay, since it's a sugar, where do we start? Where is carbon number 1? Who remembers from the very first unit? Right. It is the carbon that is clockwise from the oxygen, right? So it's that's going to be carbon number 1. So since ribose is a 5-carbon sugar, there's only 5 numbers. Again, to distinguish the carbons in the ribose from the carbons in the nitrogen base, we use a prime. Okay? The nitrogen base is always attached to carbon number 1. The phosphate group is always attached to carbon number 5. Always. Okay? So this is actually quite important. Okay? When we stick nucleotides together, okay, we always refer to the 3 and 5 ends of the DNA. So the side that ends with a phosphate is the 5 end of the molecule. The side that has an OH group is the 3 end of the molecule. So this is the 3' end, this is the 5' end. Okay? The phosphates always attach the previous 5 attach from the 5-carbon where it's attached to the 3-carbon of the next nucleotide. So the attachment is always 5'3". Okay? So DNA has directionality. Okay? And that's important. Okay. This is why directionality is important. DNA double strands are arranged anti-parallel. Okay? So the nucleotides are bound from the phosphate to the sugar between the number 5 and number 3 carbons. Okay? So the DNA molecule has a direction to it. Okay? So the two sides run in opposite directions. Right? In this one, the 3' is at the bottom, and on this side, 5' is at the bottom. Okay? So they're parallel to each other, but running in opposite directions. Therefore, they are anti-parallel. The best example of that you guys know of I can think of is the Queensway, right? You have one section of the Queensway running east and one section, the other side of the Queensway running west. They are parallel, but they run in opposite directions. The Queensway is anti-parallel. Okay? This affects how DNA gets put together. Okay? So, the hydrogen bonds between the bases are your weak bonds. The covalent bonds between the sugar phosphates are strong bonds. Okay? Why have weak bonds in the middle and strong bonds on the backbone? Right, so that you can split the two sides apart so that you can replicate. Okay? Base pairing in DNA. Okay? The purines are adenine and guanine, and they're the double ring structures. Pyrimidines are thymine, cytosine, and uracil. Okay? A's and T's have 2 hydrogen bonds between them. G's and 3's have 3. This is one way that you can dictate which base goes with which base. A cytosine can't get together with an adenine because the hydrogen bonds won't match. Okay? All right? This we already know. This was the Michelson and Stahl experiment, right? Each base pairing allows base pairing allows each strand to serve as a template. So each new strand is half the original parent and half is newly constructed DNA. So it's a semi-conservative copying process. Okay. Now, the next bit, I'm going to actually do this as a PowerPoint rather than a Google slide, just because it shows better. Biology 12. Yeah. Hi. No. I have no idea. Okay. DNA replication needs a large number of enzymes to coordinate that replication. Okay. Now, in a organism like us, our DNA doesn't replicate at one point. It replicates at multiple points. Okay. So in dark blue is the original DNA. You can see that there's a series of replication bubbles. Okay. The bubbles are where DNA is being synthesized. Okay. And these bubbles will grow until they fuse together to form 2 new DNA molecules. Okay. So in eukaryotes, DNA replication begins at a lot of different sites along the giant DNA molecule. Okay. If you took one strand of DNA out of one single cell in your body and measured it, any idea how long it would be? Yeah, it's about 2 meters in length. Okay. So that means, and think of how many cells you have in your body. It turns out if you took all the DNA in your cells and strung them end to end, it would stretch from the Earth to the sun and back. Okay. I think Hank Green said it goes around 6 times or something like this. Okay. So you have a lot of DNA. Okay. And think about that distance and how it packs into your cells, how minute it has to be. Okay. So, uh, pointer options, laser. There we go. Okay. So let's meet the enzymes that do this coordination. Okay. The first step of replication is splitting the DNA. Okay. So we want to unwind it. Okay. The enzyme that we use first is an enzyme called helicase, and it unwinds the DNA helix. Okay. So this is a replication bubble. Okay. If you're only looking at one side, it's called a replication fork. Okay. So there are two helicases inside a replication bubble, one helicase in a replication fork, and they both go out in opposite directions. Okay. You also have these light blue proteins. They're called single-stranded binding proteins. What they are there to do is once DNA gets separated, what it really wants to do is it wants to combine back together. So to prevent that, you take these proteins and you stick them in between so that they can't bind together again. Okay. And that's what these SSBPs are. Okay. They're going to make sure that the newly separated strands don't rebind. Okay. So this point is a replication fork. One enzyme that I haven't shown here, okay, it would usually travel ahead of the helicase, is one that's called gyrase. And what gyrase does is it prevents knotting of the DNA. What do I mean? If you have a long braided rope and you start unwinding the rope in the middle, what's going to form at your two ends of the rope? Well, it's going to get all knotted up because of the tension. So to prevent that, the enzyme gyrase is going to release that tension and prevent knots from forming in front of helicase. Okay, I haven't drawn it here because it usually travels further ahead than this diagram is, but it will be located somewhere off screen to prevent knotting. So that's gyrase. The other term you will hear for it is topoisomerase. Okay, there are multiple topoisomerases. Gyrase is one of them. Okay, 10 more minutes. Okay. All right. Step 2 of replication is the actual copying. It's an old biology joke. So, yeah, well, there's a lot of them in biology. Um, is to build the daughter strand. Okay, so we're going to add the new complementary line bases. Okay, the enzyme that's going to add the new base is an enzyme by the name of DNA polymerase 3. Okay, it's that line's been removed from your version. I forgot to remove it from this version. Okay, now this bit needs to be animated because I'll show you what the next couple of slides look like without animation. Right? Oops. It's already been removed. Present window. Right? You can't really tell what's going on with this image, and actually the one that's worse is this one, because these are animated slides. So if you look at them as stills or PDFs, it makes no sense whatsoever. So you have to animate it. Okay, I'm just warning you now. This is impossible to understand in this format. It has to be visual. Okay. So reanimating this. So there's DNA polymerase 3. It's going to bring in a base, and then it'll bring in another base, right? And slowly move on. Okay. So this is just going to move along and build the new strand. Okay. But in terms of what we just did, as Fillmore says, we're missing something. What were we missing? Remember that this is a synthesis reaction. Yeah, we need energy. Okay. So where's the energy coming from? Okay. Where's energy usually come from? This sucker. Right? What is this? ATP. Okay. Which is, if you think about it, a modified nucleotide. Remove the two right? If you remove one phosphate, you go to ADP. But and you get energy that you can use for something else. But there are other ways we can store energy in ATP. Could take ATP and we can remove two phosphates and make AMP. Okay. Now we have a nucleotide which can now be attached to the newly growing strand. So ATP came with its own energy to attach itself to the newly synthesized strand. And they leave behind a nucleotide. So what other energy nucleotides must there be then for this to work? Yeah, GTP, TTP, and CTP. There is also UTP when we talk about RNA. Okay, so the nucleotides, because it's the simplest one, if you look at the pathways to make all the bases, ATP is the first energy molecule probably synthesized because it's the easiest to make. We do use GTP in a few other locations, like in Krebs cycle. The one ATP that's made out of every turn of Krebs cycle is initially coming out first as GTP. GTP translates that phosphate, transfers that phosphate to ADP to make ATP. Okay, so a nucleoside is a DNA base with 3 phosphates. Okay, so that's how the nucleotides are going to come in order to allow for bonding to occur. Okay, so they all come with their own energy source. Okay, and then the energy is used by the enzyme DNA poly 3 to bind the incoming nucleotides to the growing strand. Okay, now so how do we actually add bases? Okay, this is key. You can only add a nucleotide to the 3 end of a growing strand. Okay? So, since this is already labeled 5 over here and 3 up here, this end has to be a 5 and down here has to be a 3. Okay? Now, DNA poly 3 is a little bit quote unquote dumb in the sense that DNA poly 3 does not know how to start a strand of DNA. So if you have a strand of DNA sitting there and nothing's on the other side, DNA poly 3 will simply come along and do nothing. Okay? But if you have a DNA, if you have another DNA already here, then poly 3 can attach the next DNA to the DNA that's already there. Okay? So that's why this G is here. It's a starter. It needs to be there so DNA poly 3 can attach the next base to something. Okay? Because it doesn't know how to start on its own. Okay? So the strand goes from the 5 end to the 3 end. So there's DNA poly 3. And note, right, this is the 3 end of the sugar. This is the 5 end of the incoming phosphate. Note where the 2 phosphates that are going to come off are. We'll get there. The phosphate breaks off, okay, and you create a bond. Next nucleotide comes in, the phosphates break off, you get a bond, and so on. So 5 ends up there, 3 end down there. So it always grows 5 end to 3. Okay. All right. I'm going to stop right there. I want you to review these slides on replication for tomorrow, because tomorrow it's going to get way more complicated. Okay. Or not tomorrow, Thursday. Okay. So you have a couple of days to review. Yeah, it gets a lot more complicated, unfortunately. This is just the start. That's why I'm doing it slowly and not telling you how DNA gets started yet, because I'll add to the story little by little. Yes, the third unit will have a test. The last two probably won't. I might do quizzes, like multiple choice only quizzes, but I'm not going to do a test. But this unit will have a test, yeah. Because these are the 3 base units that you need for first year university, that you really need. The other 2 are not as important. All right, 10:50, go off, study up well for your test. If you have questions, you have until 4 o'clock to ask me. All right, take care. Have a good day.